Bob Nye the Bio Guy. Bob, 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 Bob. Bob Nye the Bio Guy. Hi folks, it's Bob here, and today we're going to learn about DNA replication. The process is essentially when DNA copies itself, but we're big kids, so let's dive deep into this, shall we? This molecule to my side might look pretty familiar to all you viewers, and it should. It's DNA. This is the genetic material of life. From this molecule, called the parental DNA, we will observe the process of DNA replication, which results in the daughter DNA. There are three main steps of DNA replication: unwinding, complementary base pairing, and joining. Step 1. Unwinding! So first off, let me introduce this cool dude here. Zwam! This is DNA helicase, an enzyme which unwinds the DNA and separates the parental strands. This creates a replication fork. Single-stranded DNA is less stable, so this guy here, Zwam, is a single-stranded DNA binding protein. This attaches to the DNA to stop the helix from reforming. That's pretty much it for step 1. Step 2. Complementary base pairing. Now before we can start replicating, DNA needs to be primed. DNA primase places short strands of RNA on the DNA strand. Then this guy, boom, called DNA polymerase, comes in. Polymerase does a bunch of stuff. First of all, it begins DNA synthesis at the places where the DNA primase was placed. It forms complementary base pairs with the nitrogenous bases on the parent strand. Remember how it goes, kids. Adenine matches to thymine, and cytosine matches to guanine. Polymerase also proofreads its work for any errors. This is a nucleotide excision repair. The end error rate of DNA polymerase is only 1 in 100 million base pairs. Like, whoa, that just rocks my socks off. Here's the thing about DNA. The two strands run anti-parallel to each other because of the molecule's structure. This means one strand moves in a 5 to 3 direction, while the other strand reads 3 to 5. But DNA can only be synthesized in the 5 to 3 direction. So, what happens? Well, one strand, the leading strand, has it easy. It's synthesized in the 5 to 3 direction because the parental strand was 3 to 5. The other strand, called the lagging strand, has to run the opposite direction, so DNA polymerase synthesizes short 5 to 3 segments which start and stop periodically. These segments are called Okazaki fragments. Step 3. Joining! At this point, replication is almost over. DNA polymerase goes back to change RNA primer strands from step 1 back to DNA. Then we have this guy, shapow! DNA ligase, or ligase, whatever you want to say, which acts like a glue that joins over our Okazaki fragments together. And now you've got two daughters from one parental molecule. Isn't science great? Now, let's just talk about a couple of more things. Prokaryotic DNA differs from eukaryotic DNA replication. Prokaryotic DNA is a single 100p chromosome, so replication that begins at the origin of replication and ends when two DNA polymerases meet at termination region. In eukaryotic DNA, replication occurs at various sites, so replication bubbles spread until they meet. DNA polymerase cannot replicate the ends of the DNA, however, so the ends of the DNA are capped by these tiny little things called telomeres. Telomeres are essentially a shield for the end of the DNA. And that concludes today's episode. Tune in next week at Bob Nye the Bio Guy. Bob Nye the Bio Guy. Bob, 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 Bob.